Okay, good day everyone. In this video, I am going to explain on chapter 9, Growth, DB014, based on compressed notes. Okay, so for the first thing that you have to know is the measurement of growth. But before that, you have to know also the definition of growth itself. So what is it? It's a permanent and irreversible increase in size of an organism over a period of time. And the phases of growth consist of cell division, where cell basically divides to become many cells usually by mitosis. And secondly is the cell enlargement, where the cell enlarge or membesar, di mana kenapa dia membesar, the density of the cytoplasm increases due to the increase of organelles. Okay, and the third one is cell differentiation, di mana dia membeza untuk menjadi specific organs yang menjalankan specific function. Okay, so parameters of growth is biomass and size, di mana biomass ni consists of wet and dry mass, while the size consists of length. Okay, so firstly we are going to see the differences between wet and dry mass. So basically, what is wet or fresh mass measurement? It is the overall mass of an organism in a specified environment with its water content. Okay, maksudnya kita tak ubah apa-apa water content dalam badan dia. And while for the dry mass, it's the mass that is obtained after all of its water content has been eliminated or removed through drying. Okay, so it means without its water content lah. Maksudnya kita keluarkan semua airnya dalam badan organism itu, baru kita measure the mass without the water. Okay, so what is the advantage of wet or fresh mass? It is easy to measure, where we just take the organism and then we just measure it, and requires little preparation lah. Okay, and it doesn't cause injury to the organism, so tak perlu nak membunuh, tak perlu nak membakar organism itu, and can be used to monitor growth of organism over a period of time. So maksudnya bila organism itu tak injured, okay, so we can continuously measure the organism lah bila dia membesar. Okay, so over a period of time. While for the advantage for dry mass, it is more accurate measurement because we don't take into account the water content of the organism, because the water content of each organism is different. So we only take the cells, the dried cells that is left after drying process. Okay, so what is the disadvantage for the wet and or fresh mass pula? So doesn't measure the true growth because it may give inconsistent readings due to the different water content in that organism. So it is influence and fluctuations in water content of the body. So if the organism drinks a lot of water, so it may causes the weight of the organism or the mass of the organism increases. And while for the organism that drinks a little amount of water, it will cause less amount of mass lah for the organism. So it is inconsistent. So what is the disadvantage for the dry mass pula? Okay, so it needs to kill the organism because we have to dry the organism, remove the water content of the organism. And growth of the same specimen cannot be measured continuously. Why? Because we have killed that organism. So the growth of that specific or the same organism cannot be measured continuously. Sebab kita dah bunuh dia. Okay, can't be used to monitor growth of organism over a period of time. As I said before, dia dah mati. Okay, so the next thing that you have to know is growth curve. So for your syllabus, you only have to know about sigmoid growth curve, where it is when a parameter is measured at intervals over a period of time throughout the growth of organism. Okay, so sepanjang dia punya growth itu, dia punya parameters tu akan dikira sepanjang dia membesar. Okay, so actual or sigmoid growth curve can be plotted. Is the cumulative increase in size over a period of time, as I said before. It shows the overall growth patterns of organism and is usually sigmoid growth curve or an S-shaped curve. Okay, and is divided into 4 phases, which is lag phase, log phase, and decelerating phase or linear phase, And the last phase is plateau or stationary phase. Okay, so we go to the graph. So here basically is the sigmoid growth curve here. So as you can see, it is S-shaped and it is divided into 4 phases: lag, log, decelerating, and stationary. So graph is obtained by plotting growth parameter y-axis against time x-axis and can be used to describe growth of organism or population of unicellular species. So kita boleh tengok dia punya populasi organism itu, contohnya adalah paramecium. The number of the organism in a certain period of time. Kita boleh kita boleh nampak menggunakan absolute growth curve ini. So shape of curve is sigmoid shaped and it is divided into 4 phases. So we go to the phases. So lag is basically maksudnya lambat. Okay, dalam bahasa Melayu dia lambat lah. So it's a slow growth because cell divide and cell enlarge. Masa time ini cell akan banyak-banyak kali divide by mitosis and membesar, menambahkan dia punya organelles. Specifically dia akan tambahkan banyak mitochondria. Okay, so cell division and cell enlargement occur at slow rate. Organism is young and unmatured and start adapted to a new environment. Maksudnya dia masih adapt to the new environment lah. And bila dia dah adapt, dia dah masuk log phase di mana growth akan proceed exponentially. The rate of the growth increase and proportional to the number of cells that are present. So there are no limiting factors. Sebab kenapa there are no limiting factors? Sebab that organism dah adapt to that environment. Dia dah tahu berapa suhu, berapa makanan, apa jenis makanan yang ada dekat kawasan itu. So there are no limiting factors. So the rate of growth is maximum and the condition of is optimum with sufficient food, space, oxygen and no accumulation of waste products. So masa ni masih menambah-nambah lagi lah populasi organism itu. Okay. Tapi tak berkurangan lagi dia punya food or space or oxygen pada masa itulah. And sampai masa phase yang ketiga, decelerating or linear phase, di mana growth rate becomes limited and slow down. So rate of growth takes place at constant rate, okay, because it is limited by internal factor. Maksudnya maturity or genetic, maksudnya dia punya spesies itu hanya time situ saja. Tahap itu je dia punya pembesarannya. Okay. Or external factors di mana adalah limitation of food dah berkurangan, space pun dah sempit, and ada competition untuk berebut makanan yang ada ataupun berebut mate ke, berebut tempat ke. And the last phase untuk the sigmoid curve adalah stationary or plateau phase. So relatively constant growth di mana net growth equal to 0 as the rate of cell division equal to rate of cell death. Maksudnya sel yang mati tu sama dengan sel yang baru hidup. So dia punya rate tu, net growth tu masih akan kekal 0 lah. Okay. A period where overall growth has ceased or stopped. Okay. So size of organism unchanged. Dia tak berubah. Dan populasi pun tak akan bertambah. Dia akan bertambah. Tapi bila dia tambah tu, yang mati pun sama je. Okay. So the organism has achieved maturity and overall growth ceases stop. And the rate of cell division equals the rate of cell mortality. Sel yang baru divide, yang baru sama juga yang dengan sel yang dah mati. So for this chapter 9 DB014, that is all that you have to know. So the first one, measurement, done. And the second one is the growth curve, which is sigmoid growth curve, done. Okay, so thank you.